Okay, unknown number 6 will be the enteric organisms. We'll be looking at a group of organisms that will include Pseudomonas aeruginosa, Klebsiella pneumonia, Enterobacter cloaca, Citrobacter freundii, Escherichia coli, and Salmonella species. These are all gram-negative rods, and they're all pretty much the same size and shape, but we will want you to inoculate some selective media that will help you in the identification of these. The first one will be McConkey agar, which you've already been exposed to. You know it only grows gram-negative bacteria, and only those that are lactose fermenters will produce a pink colony on this agar. There's another medium that we're going to give you in this lab called Hectone Enteric agar. We don't have an example of it today, but it'll be kind of a blue-green colored agar that's, again, is selective and differential for these enterics, and you'll get a kind of a salmon-colored colony that will indicate, again, lactose fermentation. So you're going to be inoculating a lot of media with this lab that you're already familiar with. A couple we'll be introducing to you, but you know the TSI slant, you'll be inoculating that. The motility S agar, you've already been exposed to that. A couple of new ones will be citrate agar, so if you inoculate just the slant only on this citrate agar, and after 24 hours of incubation you get a bright blue color produced, that will indicate citrate production. And then also a real important medium with this group is the Indole Test, and we do the Indole Test in tryptone or tryptose broth. You'll inoculate this broth just like we inoculate the phenol red, just by putting your inoculum in there and twisting it around and just inoculating the broth. This will then be incubated overnight, and we'll come back after 24 hours and we'll add a reagent to this called Indole Reagent. And the Indole Reagent will detect, Kovacs reagent will detect indole production in the broth, and you'll get a red ring forming around the top of the medium. So all of those will be inoculated, and you'll be incubating 24 hours and coming back and reading your results the next day. Okay, so to perform the oxidase test for cytochrome oxidase, we're going to use a piece of filter paper, and we're going to put a drop of oxidase reagent on that paper and allow it to soak in just for a little bit. Then we're going to take an isolate from an isolated colony on a plate and streak that right in the center of that drop, and we'll look for blue color to develop within 30 seconds. So if it turns blue like we see here, that's a positive reaction. You have a negative? Well, a negative would be just like nothing. Mm-hmm. So no color reaction within 30 seconds is a negative.
Okay, so this is the slide test for the Salmonella agglutination. Placing a drop of sterile water onto the slide We're going to add our bacterial colony to it and make an opaque suspension. Then we're going to take our Salmonella O antiserum, place it next to the bacterial suspension. Using a wooden applicator stick, we're going to mix these together. Once they're mixed, you can kind of rock the slide back and forth. And if it's Salmonella, it will agglutinate. And if it's not the freshest Salmonella, it will not agglutinate. So this test needs to be performed within your 24-hour incubation time on your blood agar plate to get a proper reaction. And it's starting to agglutinate. It's starting. It's slow. That's our test. So we're going to take a colony from our blood agar, using a sterile swab, and inoculate our saline. We want to do this a little bit at a time, and we don't want it too opaque because we don't want to have a completely overgrown plate. So doing this a little bit at a time, we'll compare it to our McFarland tube over here. If it's focused or not. And so they look about the same in the light, which tells me I can swab the plate. So now that we have the tube to how we want it, our suspension, we're going to put a new swab in there. And we're going to create a long You don't want to dip back into the tube. You want just a nice, even, thin layer of bacterium. And when you're doing this, it's a lot easier to see where you're streaked versus where you haven't streaked yet. I don't know if that's coming up on the video or not. There we go. Now that we've created our long, you can see it's completely streaked.
That's good.
Once you create your lawn, you just kind of let it air dry a little bit. And then we have our handy dandy automatic dispenser. And then you can tap down very gently the disc just to make sure they're there on the agar, because we're going to incubate this inverted. And that's it.
This is the Indole test. In the rack in front of us, we have a culture that was incubated for about 24 hours in tryptose broth. I am adding 5 to 10 drops of Kovacs reagent, and as you can tell by that dark red, hot pink color change, we have a positive Indole test, indicating that tryptophan was hydrolyzed by this particular bacterium. I am adding 5 to 10 drops of Kovacs reagent to the broth. There is kind of a tan discoloration, but this is just what the Kovacs reagent looks like. So this would be a negative result.